PhD of neurobiology from University of Science and Technology of China in 2004. He then received 2 postdocs at the University of Toronto and afterwards in the Harvard Medical School. And he then became professor of neurology.
Rosa, is that she's
the audio stopped, right?
We lost Claire.
Yeah, we lost her.
I'm here. Do you see me now?
Yes.
Yes.
I think my connection was lost. I'm sorry. So he was professor of neurobiology and associate director for neuroscience PhD program at Mayo Clinic. And he's now currently in transition to become full professor and founding director of Center for Neuroimmunology and Glial Biology at the Institute of Molecular Medicine at University of Texas Health Science Center at Houston. And he is also a C. Harold and Laureen G. Wallace Distinguished University Chair. And Dr. Long-Jun WU, is research primarily focuses on the neuroimmune interaction, particularly in the function of microglia in normal and diseased brain. So the floor is yours. Sorry for the interruption.
Oh, no worries. Thank you. Let me share the screen first. Let me share the screen. So you can see my screen, right? Yeah. Thank you again for the introduction, and thank you for inviting me to this platform. I really enjoy this platform for years now, and I'm glad Rosa and Amanda, you guys, the team, continue this amazing platform after COVID. Yeah, today I would like to share our main research interest for lab, talking about neuroimmune interaction, particularly focusing on how microglia sense and regulate neural activity. And just to mention about the transition. Indeed, I am still in transition from Mayo Clinic to University of Texas Health Science Center at Houston. So my lab will be closing down in September in Mayo Clinic, but we start a new center called Center for Neuroimmunology and Glial Biology in Houston. So really exciting, like a new center with a focus on neuroimmunology. So the long-term mission here is that, I mean, everybody in the field, we're talking about how we use our immune system, brain immune system, to treat brain diseases. That's the kind of long-term mission, right? Idea really, we are borrowing the main breakthrough in cancer immunotherapy, how we use immune system to battle cancer. So similar idea, we have brain immune system, and how can we harness the power of our brain immune system to treat brain diseases. So it's a new center started this January, although my official appointment started in March, and we are recruiting faculty and all level of research. It's like postdoc student as well, but we do have openings, multiple openings, assistant professor, social professor level. So anyone interested, shoot me email. So I think for this audience, I don't need to introduce microglia, but I'm not interested. So microglia are very unique because they're dynamic. So my first paper was published many years ago now. So it's using imaging approach to study microglia dynamics and later microglia-neuron interaction. So you can see microglia itself, like cell body is not moving much, but they're using this multi-process to survey the brain environment. And when there's like ATP mimic damage, so microglia was dramatically like sense to that and send the process like towards ATP. The cell body not moving much, but mostly the process, particularly is acutely. So we keep using the imaging approach to study this dynamic microglia-neuron interaction. So here I can show you like what we can use in vivo image 2-photon microscopy to image microglia in green, G6 and GFP label, and then neuron with SAR1 YP label. So you can see this from surface to all the way to 500, 600 micrometer of the brain. we can see this intimate interaction between microglia and neurons. So in my lab for many years now, so we focus on microglia-neuron interaction at a different level, like a cellular level, what's the signaling mediate this interaction, and then at circuits level, how microglia integrate in neuronal circuits. And then in our lab, we use many different disease models, including pain, seizure, neurodegeneration, autoimmune disease model to understand in disease context how this microglia neuron interaction may alter, and eventually how we can target microglia or neuroimmune mechanism for treating brain diseases. That's kind of the theme for my lab research for many years. So for today, I will mainly focus on this basic question about how microglia interact with neuron. Particularly, I will talk about ask two questions. The first is how microglia sense the environment, in particular about the neural activity in the brain, and then how microglia can engage into neuronal circuits and the neural activity. So the lab really start the first really straightforward experiment many years ago when Al, my former postdoc Al, was in the lab, is that we use brain slice preparation. So we ask a very simple question when we perfuse glutamate, right, to mimic seizure causing neuronal firing. So what happened to microglia? Right. In the seizure context, if you read literature, people already talk about microglia get activated, usually talk about weeks and days, you'll see microglia being amoeba shape. But if we use imaging approach, we are talking about acutely, like within minutes, right? Here, we again label microglia with GFP, neuron with YP. So you can see this is a minute, right? Within like this is a glutamate application. So within few minutes after glutamate application, so we found microglia send the process interact with neuronal cell body, interact with neuronal dendrite. So really we found it's striking microglia acute response to neuronal hyperactivity, that sending process towards neuron. We name it microglia process extension. So the lab really start from that work. This is our first research article published from lab. Really, we about 10 years ago now. So I'll just show you the conclusion is that what we found is this: when we activate neurons, so there's MD receptor dependent, calcium dependent, this release of ATP. Remember, I showed that microglia super sensitive ATP because of the P12 receptors. So when hyperactive neurons, they can release ATP and detect by microglia. That's why you see microglia sending process extend to neurons. So that's kind of the first research project we started, and very straightforward. And then we along this line, we also try to see like if we directly manipulate neurons, what happened to microglia. So here we can use recording, patch clamp recording, just do the single neuron like activity. So we can inject current, fire action potential, and then we can inject dye into that neuron, so we can trace axon and dendrite, and in the GFP microglia GFP mice, so that we can trace when we activate neurons what happened. And we also found a very interesting phenomenon that in microglia neuron interactions, microglia can send the process converge to the hotspot of the dendrite or axons. So this in the brain slice, if we do in vivo, we can use seizure models, like use a kinase acid injection model, so induce seizure, and in the cortex, again, we see this very interesting microglia process convergence. So here the neuron is the layer 1, so the neuron dendrite was labeled with like a YFP and then microglia GFP. So you can see in vivo context, microglia also send the process convergence to the hotspot. We name it microglia process convergence. And also, if you look carefully, so you will see this pouch-like structure. So those are the swollen dendrite, and microglia actually form the pouch along those swollen dendrite. So they're quite stable there as well. So it's another form of interaction. We name it microglia process pouch, and also in the seizure context. So for many years, so we were studying this microglia interaction with neuron when neurons hyperactive. So the idea we started that question is just because to start a communication, we want to amplify the signal. That's why we use seizure as a model and to make a hyperactive neurons. So for many years, we study this and we found ATP P12 is one of the major factors released from hyperactive neuron or the environment, probably exercise as well, and then sensed by microglia and that mediated by P12 receptor and the interaction. So we describe a different type of interactions, and then later I'll show you like what this means, that interaction means to neural activity, and then we largely believe this interaction is neuroprotective or like in relatively acute stage. So this line of research really was led by my really talented postdoc, former postdoc Upcon Ao. Now he's a social professor at University of Virginia. At the same time, we also ask like similar question. Now we want to ask about how about the hypoactivity, right? So we know microglia is super sensitive. So how about in the environment that the activity is shut down? For example, here I just show you this is a GCamp recording of neuronal GCamp, and you can see in the awake context, this is a layer 2-3 neuronal layer. This is layer 1, the dendrite layer. So you can see neuronal activity was very obvious, right, in the awake context. But then when we use isoflurane or other anesthetics, we can shut down the neuronal activity. Like this brain environment are totally different. So we ask a similar question in these contexts. So what happened to microglia, right? And an idea here, after many years of study on epilepsy, we think the microglia follow neural activity. So the prediction at that time is that when we shut down the neural activity, we can shut down the microglia like dynamics. But surprisingly, what we found is that when here's only microglia imaging now, so labeled with CXCL and GFP. So you can see in the awake context, microglia, this dynamics basically is like a restraint. And then during anesthesia, we found the microglia extend the process and increase the dynamics. This is very surprising, and we can do simple analysis. Neural activity can shut down. This green line shows the neural activity immediately by anesthetics. But when we look at microglia process area change or the surveillance territory, they'll gradually increase, even the velocity increase as well. When we look at morphology, and the same thing, like because they extend the process, so we can see they have a longer process. Also, for the short analysis, show they have more complex morphology as well. So again, indicate that during anesthesia or during neural hypoactive context, right? There's also increased microglia process dynamics. The question here then is that the neural activity or is anesthetics, because anesthetics can do many things. To answer that question, so we can use optogenetics, can locally like reduce just neural activity. Here we can express channel rhodopsin into GABAergic neurons, and then we can use a light to shut down the neural activity. So basically, it's just only manipulate neural activity now. So when we lower the neural activity in the cortex, and when we look at the microglia dynamics same time, and again, we found there is increased microglia dynamics, process area increase, or territory, surveillance territory increase in the optogenetic manipulation inhibition mice. So suggest that when we just use optogenetic to inhibit local neural activity in the cortex, we can also increase the microglia process dynamics, indicate this is controlled by neuronal activity, right? So then the question becomes, what's the mechanism mediate this increase of process dynamics? So we go with P12 again, because this is one of the most important receptor control the microglia dynamics. And surprisingly, we found that in P12 knockout, this process dynamics still increased in the knockout mice. So just this is not a P12 dependent mechanism. So to further answer the question, what's the molecular receptor mechanism mediate this? So we did a small functional screening. So idea here is that during this anesthesia, maybe there's a lot of neurotransmitter reduced, right? Maybe one of these or multiple of this could mediate this increased dynamics. So can we add back, right, neurotransmitter one by one to see which one can inhibit this increase? So that's the idea. Very luckily, we found the NE is one of the major player here. So when we add other neurotransmitter like serotonin, acetylcholine, or dopamine, none of them can reduce this increase, right? But when we add NE, so we can almost abolish this increased dynamics of microglia and anesthesia. So suggest the NE is the one that control the microglia process dynamics. So this also published a few years ago now, so I will just go to this main conclusion, and I skipped the data about the beta-2 receptor. And what we found, the microglia have this beta-2 receptor, and the beta-2 receptor can control or restrain the reduce the microglia dynamics. So in a awake context, there's a tonic NE level. So through the beta-2 receptor in microglia, that can restrain the microglia dynamics. But then during anesthesia, now there's reduced NE tonically in the environment. It's more like disinhibition now. That's why now microglia can will increase the dynamics. So this is a basic idea. So this work was mainly carried out by my postdoc, a former postdoc Yung Niu. So now he's also a social professor in South China University of Technology. And we now like I would like to just mention about this because it's a really exciting like we published together with Annie and Majestic's group, and I want to give credit to her as well. So it's we also, if you look at the title, very similar. So it's quite striking. We when she put in the bioarchive, and we found out, and we have to put in this almost the same day, next day in the bioarchive, and eventually published back to back. And I want to mention is also because we use different physiological contexts. I did not show data here. So in our paper, we use a whisker trimming as a physiological context to reduce neural activity. So then we measure microglia dynamics in the barrel cortex and increase microglia dynamics. And in Annie Majestic's group, and they use visual cortex as a study microglia dynamics, and they cover the eyes to reduce neural activity. They also increase the microglia dynamics in the visual cortex. It's kind of like a synergistic effect. So now I try to summarize this part is that what we found is we come up with this U-shape response ideas that microglia dynamics, they respond to neural activity in this U-shaped response. So, and the hyperactive context like seizure, so microglia can sense this neuronal hyperactivity through ATP-P12 dependent mechanism and increase the dynamics. But during the anesthesia, like hypoactive context, microglia also increase the dynamic through this NE beta-2 disinhibition mechanism, right? That's kind of the way we are thinking how microglia can sense neuronal activity. So now what I just show you is more using microglia structure dynamics to process image microglia process dynamics as a readout. And now I want to show you, we are also using another readout, that's the microglia calcium as a readout, activity as a readout to see how microglia can sense the environment. So here just show it's more like a positive control, so we can express GCAM6 in microglia, so we can use laser burn, so you can see the microglia have a very nice the activity, calcium activity. So this is another readout to see how microglia can sense the environment change. And to answer that question, now we can also try to precisely manipulate neural activity here, use a chemogenetic approach. Here we can express the GQ dread or GI dread into excited neurons. So that way we can use the CNO to precisely increase neural activity, or in GI dread case, we can inhibit neuron activity. So what we found interesting when we use GQ drugs in excited neurons, so we use CNO to increase neuron activity. What we found, the microglia calcium is increased mostly in the process, not in soma. So the green line shows the soma, and the soma neuron microglia calcium is not increased. But when we look at the microglia calcium in the process, so there's a dose-dependent increase. And also interestingly, when we inhibit neural activity, here we express the GI drugs, right, chemogenetically in neurons. And we found that when we decrease neural activity, we also see the microglia calcium increased in process, but not in soma either. So showing that this, again, this kind of U-shaped idea. Here I just show you the movie, and this is a GQ drug context. This is GI drug context. When we add CNO, so you can see the microglia process calcium both increased. You will see this morphology not as beautiful as a GFP, just because of the microglia calcium mostly happening in the process. So so you can see the in both neuronal hyperactivity and hypoactivity context, right? Microglia calcium increase, particularly in the process, and we actually also did isoflurane and seizure, and two extreme cases as well. So the increased neural activity in seizure context, or shutdown neural activity, isoflurane in any seizure context, and then also GR drug or GQ drug precisely control neural activity. This is the neural activity change. Then at the same time, when we look at the microglia process calcium, we again see this very interesting U-shaped response. And we are still working on the molecular mechanism of this, how calcium increased. For example, recently we found that in the seizure context, actually UDP P2I6, that's the one of the major mechanism mediate the microglia calcium increase during the seizure context, and we were still working on this side. And this line of research mainly carried out by my postdoc. Actually become formal postdoc a couple of days ago. He started his own lab now as assistant professor in University of Minnesota 2 days ago, three days ago. So anyway, so this is we are using the calcium imaging, the microglia calcium as a readout to study how microglia sense neural activity, and again we found this U-shaped response. So so now I would like to talk about the next question. So the question about how microglia can engage into neural activity, right? So I will show this microglia that can sense both hyperactivity and hypoactivity. So what they do, right? So back to our very original paper, talk about microglia process extension interact with neurons, and we found that microglia, they can, when they interact, seems like they have this bulbous ending structure with neuron. And also when we use P2R12 knockout, this we know that will reduce this interaction. We found the seizure is much stronger in the knockout mice. Here we use ICV kinase seizure. We also use different type of seizure model like IP kinase. We see the similar effect that without P2R12, without this interaction, so the seizures are largely increased. So this data suggests that microglia may be able to dampen neural activity through the P2R12 dependent mechanism. That was like our first research article on this. And then like Adams Denise papers very beautifully show that indeed there is structure basis for this interaction. So they also show this bulbous ending, but they use super resolution microscopy to show this is the structure basis. Again, this is P2R12 in the microglia, but also in postsynaptic. There's an enriched potassium channel there, and they name it somatic purinergic junction. Those is a structure basis of this interaction. And also in his paper, he beautifully showed this direct evidence. Here they use P2R12 inhibitor instead of knockout. They they use inhibitor when they inhibit this neuron microglia interaction, and then when they did a calcium imaging in neurons, so they show this increased neuronal activity. So there's direct evidence to show indeed this is a P12 dependent like a microglia damping neural activity. And then in our paper and Adam Denis paper, we still don't know the molecular mechanism. And then Anne Schaffer's papers group and show beautifully there's a mechanism behind this it's dependent on the adenosine mechanism. It's like a microglia not only use P12 to go to interact with neurons, but also have this ACT enzyme like CD39, CD73 to degrade ATP eventually to adenosine. And we know that adenosine is able to inhibit neural activity. So give a very nice mechanism how microglia could dampen neural activity. So so in her study also they use P12 knockout different seizure model and also show there's in the P12 knockout there's increased seizure as well. So overall we largely agree that when there is neuronal hyperactivity, there's increase in microglia neural interaction, they're able to dampen neural activity. So then we ask a question like, how about this microglia function, right, during the neuronal hypoactivity? As like I mentioned right before, when we have a neuronal hypoactivity, we see this increased dynamics as well. So here to show you again, when there is like a awake context, microglia process largely restrained, and then during ice flooring, now you see the microglia increase the dynamics, and then this increased dynamics can also largely like maintained for hour, like couple of hours. So suggest that there is a specific like a microglia dynamic change, but also can be lasting more than just the neural activity. So more interesting when we look at this microglia more closely about this increased dynamics, we also found this increased Bob's ending like structure in the microglia process ending. So you can see the norm. This is a awake context. Now in ice flooring context, so we will see increased microglia processes. Magenta show that. And then when we focus on zooming those endings, so we can see this is a Bob's ending like it's enlarged ending. So we can do the calculation about this Bob's ending, and this Bob's ending can maintain for like around like 30 minutes of like 1 hour during emergence, after recovery of the anesthesia. So you can see this is a summary result. So during the ice flooring and during the emergence, so there's a increased not only microglia process dynamics, but also the increased Bob's ending structure. So the question become like, what this Bob's ending structure do, right? Obviously, we want to look at what they do with neurons. So here we can imagine neurons with viral labeling TD tomato, and then we found that microglia increase the dynamics and actually increase their interaction with cell body, and this increased cell body can maintain and during the emergence, and also they increase the interaction with dendrite as well, and seems that this increase in interaction with dendrites now maintained, so recovered during the emergence, right? So suggests that when there is microglia process dynamics, when there's a Bob's ending, the seems like it's a increased interaction with neuron soma and the dendrite. And here's a movie to show that. So neuron labels T tomato or cherry, and then during ice flooring, so you'll see the microglia have this Bob's ending structure interact with a neuronal cell body, and this can be maintained during the emergence. And then we really want to see like what they have this Bob's ending onto the neuronal cell body, what they really do. And of course we have to go to the like synaptic level now to see what happened to the neural structurally how they interact. And I will skip the glutamatergic data, but I want to show you the main conclusion is that what we found this microglia bulbs ending to the cell body mostly overlapping with the VGAT GABAergic terminal. So here the VGAT in red showing the GABAergic boutons onto the cell body. This is the cell body of the neuron, excitatory neuron. So microglia labels IBA1. So you can see the microglia process ending, bulbs ending, mostly interact with the GABAergic terminal. So these interaction and normal context, so not much interaction. And then during the ice flooring and then even during the recovery emergence, so you can see this is largely increased. And here just show the contact also increased during the ice flooring context and the emergency as well. So showing that when we found the microglia increase the dynamics and the increase of neuron, what they do is that increase interaction mostly with GABAergic boutons. So to study this more closely, because this is a confocal imaging, so we really want to go to the more detailed interaction. So we use the super resolution microscopy to see what exactly they interact with boutons, PV boutons or GABAergic boutons. So here we found that interestingly when microglia interact, so you can see the microglia process seems like they can have this close interaction, but also insert into the synaptic like a cleft. And we later name it shielding because they really go below between the boutons and the cell body. So it's really a very interesting observation. And we're not satisfied about this resolution, really, like what exactly they do there. So to further answer this question, so we want to correlate this like light microscopy with EM. So here we can use a 2-photon microscope, right, to image functionally how microglia interact with neurons, interact with the PV bouton. The PV bouton is labeled as virus. So then same time we can actually we have a GCamp in neurons as well, so we can image the neuronal activity in the same neurons. But in that 2-photon imaging, we can laser brand that area, and then we now go to the EM level to find exactly same interaction in the EM. So it's a very challenging experiment by my postdoc Koichi really did great work to solve this problem. So we can see indeed when microglia interact with neurons, the process ending, they basically like insert into this synaptic cleft. And we name this shielding, synaptic shielding, and then here's 3D rendering. You can see the microglia basically have this dynamic process they can insert into the synaptic cleft. So what we found interesting later is like during the emergence, right? They can still they later still recover, and after recovery, we found this GABAergic synapse still there. So suggests it's not synaptic pruning or synaptic annihilation under this context. This is more like a reversibly event. So or more like functionally blockade of a GABAergic synapse. That's the idea behind it. So the next question become like when we see this interesting phenomena of the synaptic shielding of inhibitory like inputs, right? So what they do. So as I mentioned, we were like studying, we were using GCamp neuronal activity to study neuronal activity anyway. So we want to see what happened to neuronal activity. So here it's a GCamp neurons, and we can see during the awake context, right, and then during ice flooring, we almost completely shut down neural activity. But interestingly, during the emergence, right, once the recovery, we found in most neurons there is overshooting of the neural activity. So there is most neurons have a hyperactivity compared with like a control context. So this is isoflurane, and then in those same neurons, so you can see there's an increase in neural activity. So we did color coding. So most neurons in red increased neural activity during the emergence, and some neurons didn't change, some even decreased. But overall, we found that neurons have this hyperactive phenomena. So compare with the control context, awake context during the emergence. So suggests that there's a phenomenon there. So we want to know whether the microglia, the one where we show the microglia synaptic shielding, because it's more selected to the inhibitor neurons, whether that mediate this effect, right? To answer that question, obviously we can use like a ablation approach, right? Here we can give a yes or no answer, right? We can just get rid of microglia to see whether this phenomenon is still there. So this experiment is well powered, because so we have a control context, and in the ablation context, we can image the same group of neurons. So that same group of neurons in the presence of microglia or without microglia. So what happened to their activity during the emergence, right? So what we found interesting indeed, like when we can see there's before ablation, the control context, there is increased neural activity, right? And or readout, and then after ablation, the same group of neuron, now the neuronal like hyperactivity during emergence is not happening anymore. So suggests that this indeed microglia is the one really play a key role here to regulate this neural activity. More specifically, they promote the neural activity maybe through this synaptic shielding. So to further answer whether this indeed is a microglia like dynamics, right, control. So we go back to the beta-2 mechanism, because we know the beta-2 receptor is the one that really control the microglia increase the dynamics during the anesthesia. So here we use the conditional knockout or knockout the beta-2 receptor in microglia. So under these contexts, now without beta-2 receptor, this increase in microglia process dynamics is abolished, right? So no increase in microglia process dynamics now. And then in these mice, we can now do the similar experiment, and also we can have well-powered have its own control, because we can image the neuron before tamoxifen induction, because it's inducible like a knockout, right? So before the knockout microglia, and then after like tamoxifen induction, and weeks later, right, now the beta-2 mainly knockout now, major knockout now in microglia, we can image the same group of neuron to see what happened to the microglia neuronal activity during emergence. So it's the chronic imaging. It's like more than 10 weeks imaging. It's quite challenging, but we managed to do that. So again, we can see that during before conditional knockout, and we see this increased neuronal activity during the emergence. Now after tamoxifen induction, weeks later, now in the same group of neuron, and the surrounding by the microglia, now without beta-2 receptor, now we found that those neural activity were now largely reduced. So again, suggests that this really is a beta-2 mediated mechanism, and also suggests that this is a microglia control mechanism. So in summary of this study, so what we found is that during the anesthesia, there is increased microglia dynamics, and we still don't know the detailed mechanism why they will attract to GABAergic synapse, but what we found that they can be selectively targeted to the GABAergic bouton and form this interesting bulbous ending, but also have this synaptic shielding function to block the GABAergic synapse transmission likely. And then because of that, so they can promote this neural activity during the emergence time. And this is also an NE-dependent mechanism. So when we in a knockout, we don't see this increase of neural activity during the emergence as well. So really, it's another way to think about how microglia can regulate neural activity here. we specifically found that microglia is able to promote the neural activity and also to reversibly. So like so it's not through the eliminate of GABAergic synapse, but basically it's a functional blockade of the GABAergic synaptic function. And so in summary, for what I discussed today is that well, we found this U-shape response of microglia, how microglia sense neural activity, and we found microglia can sense both hyperactivity and hypoactivity through different mechanisms. In the hyperactive context, ATP-P12 is a major mechanism. In the hypoactive context, NE beta-2 are the main mediator for the how microglia sense neural activity. But also microglia are able to regulate neural activity. So during neuronal hyperactive context, so microglia are able to dampen neural activity, likely through the adolescent mechanism, but also we also work on different potentially other mechanisms as well. So how microglia could dampen neural activity. But also what we show here, the microglia is able to like promote neural activity when a neuron active like activity is low. So it's more like through the disinhibition mechanism, synaptic shielding mechanism to promote neural activity. So they overall they can sense and regulate neural activity to maintain the homeostasis. So that's the main summary here. In the words, we also use the calcium signaling as a readout, also show this U-shaped response of microglia to neural activity. And then we found the microglia able to regulate neural activity to maintain brain homeostasis. With that, I would like to thank my lab members. I already mentioned their name, particularly my former trainees. Now they start their own lab. Afghan AO mainly work on the microglia seizure studies, and then Yong Liu mainly work on microglia, how microglia sense the anesthesia, and then Tony mainly use the calcium imaging like readout to study microglia neural interaction, and then my current postdoc Koichi is mainly work on the microglia synaptic shielding mechanism. I'd like to thank my collaborators, particularly Dr. Greg Oro. He's a physician scientist that work on the seizure model. So we have several grants together on the microglia function in seizures. And this is my current support AI most from NIH. With that, thank you everyone for listening, and happy to take questions.
Okay, thank you, Dr. Wu. It was an amazing talk. For the question and answers, for the questions, you can write the questions on the chat, or you can just write your that you have a question, and we will give you we will open the mic for you. The first is from Zainab Ilal. Can the professor share his email? In the future, I want to contact for if they have postdoc position. I'm sure you can check his papers. His email is on the corresponding section of the paper, so you can check his email right there. The first is from Alain Vessy. Thank you for this great talk. Do you think that these new mechanisms that you identify upon hypo or hyperneuronal activity are also involved in basal activity?
That's a great question. I mean, in the normal context, I'm sure the microglia is able to engage, but that probably it's not easy to study because I mean the signal is not amplified. So I don't think we know that for sure how in a very normal context how microglia is engaged, but we know microglia and have play a physiological function, like what Wenbiao showed that is important for learning memory. So it's some type of more physiological context with more activity or plasticity, microglia may be involved as well.
Okay, thank you. The next is from Mohit Dubey or Dubi. You can turn on your microphone.
Yeah, hi. Sorry for no image. Beautiful talk. Thank you for the fascinating results. I have two questions. First is about the perisynaptic shielding. I was wondering where and what kind of signals. So is it presynaptic, postsynaptics, and which signals does microglia sense to come between this cleft? So what kind of signaling and from where this signal is coming from? And the second question is, have you looked at the calcium signal when this shielding is happening that's within the microglia?
Mm-hmm. Great question. Yes, that's indeed like what we want to address. Both questions are important. So the first question is that what signal drive microglia go to the like GABA synapse, right? So in the beginning, we think maybe the GABA is the one because the microglia do have a GABA B receptors.
Right.
But
Yeah.
interestingly, when we put GABA, if you put ATP, microglia go there. When we have a GABA like pipette, microglia now going to the ATP like grading. So suggest GABA grading. So suggest that really it's not a direct sensing the GABA. Maybe something else. So we still don't know yet from the GABAergic synapse that that can be sensed by microglia. For the second question, like calcium, yes, I mean that's something we're doing right now. So we assume that there's something will happen in the process as well, because those are the bulbous ending and also the microglia calcium most happening in the processes. And the GABA B receptor, although it's GI coupled, right, it's more like P12 is GI coupled as well.
Right.
We also link to the calcium. Yeah, those are under investigation right now. Yeah.
Thank you.
Yeah.
Rosa?
Yeah, thank you. Fantastic talk, was really amazing. So in the same line of questions, so what attracts microglia to the GABA, I was thinking, do you envision a system, because I guess they need to sense hyperactivity first to shield the inhibitory, otherwise, so it's a mechanism to block hyperactivation away, so they are blocking the inhibition, they're suppressing inhibition, but they don't do this normally. So the reason why they go there can also be combined with other signaling. So do you think that they sense this like unusual state of neural activity to target the inhibition, to relieve, to suppress?
Yeah, that's very likely, right? So we're thinking even other cells may be involved, maybe not directly, like because all those synapses, and like if you talk about astrocyte, right, the astrocytes everywhere. So could be the astrocyte also have this, astrocyte also follow neural activity as well very closely. So when there's lower neural activity, not only the GABAergic, the synapse, the GABA released, but also local astrocyte may sense that and release something that the guide microglia go to the synaptic cleft. So yeah, definitely some other factors, unknown factors involved. So still a long way to go to find those out. Yeah.
And also linked to this, because you said in the talk that this is reversible. So it's a shielding that is reversible. Is it this a function of time? So if you prolonged the isoflurane, for example, state, is it still reversible? Have you tested this as a function?
That's a great question. That's something like I talk on a postdoc. We should test that. Yes, I mean, for prolonged, there's in clinic also there's they talk about this anesthesia induced memory deficit. Usually it's prolonged and also combined with surgery or these things. So next step, we definitely need to do that. So more precisely to control the anesthesia and also combination with more clinical relevant context to see how this interaction may be altered and what's the detailed mechanism there.
Thank you.
Okay, Amanda, she doesn't have the microphone, so I will make her question. Actually, she said that we can leave her question for the last, but actually it's very related to the question from Rosa and the previous question. So she asks, what do you think drives microglia towards the GABA synapses? I am thinking of an old paper by Wei T. Wong in the retina showing that the response to GABA was mediated by ATP.
So that could could be true as well. I mean, there's recent Bo-Peng's paper and also Zhihua's paper show that ATP somehow is also involved. We have unpublished results, so we did some ATP imaging as well. In the anesthesia context, somehow that means there was a ATP hotspot release. So but those, I doubt it because the ATP is released, the hotspot is quite sparse. So I don't believe that will be just around that synaptic shielding side, but that's something we should test in the next steps. Yes.
Okay, the next question is similar, so I will read it, but it's been already answered. So it is known what attracts microglia to GABAergic synapses to create the shielding. Do you think it's not epinephrine directly? And the next one is a great talk. Did you test the response of mice to anesthetics after microglia ablation? Any behavior change appeared?
Yeah. The first one about this NE, yes, I think the NE is involved, but may not be because NE is overall, we did also NE use collaborate with Lee's group. We also have NE sensor to measure the NE. So NE is more like more broadly change. So I'm not sure that will also specifically target to GABAergic synapse to mediate that, but the NE overall can control the microglia dynamics. That's why we see the phenotype in a beta-2 knockout. Again, we need to find out the specific mechanism how microglia is tracked to the GABAergic synapse. So in terms of the behavior, yes, we did. I actually didn't have time to show here. So we did some behavior analysis. They have a behavior sensitization during emergence, just like if you more like one Fred test the pain behavior. So the microglia is more sensitive to this sensory touch during the like emergence of anesthesia. And we found when we ablate microglia, we also ablate this hypersensitivity. So suggest that this neuronal hyperactivity controlled by microglia can be translated in some degree on a behavior level as well.
Okay, the next one is from Vane Steinhoff. She wanted to ask if there is a sort of maturation of the behavior of microglia towards GABAergic neurons. Describe it in your talk during development alongside the switch from excitatory GABAergic to inhibitory GABAergic neurons.
That's a great question. So we haven't tested that, but interestingly, like there's a report on this microglia is able to displace GABAergic boutons during the developmental stage. That is important for damping neural activity during the seizure because a developmental seizure. So during development, right, GABAergic synapse are more like excitatory, right? So microglia seems like can insert cell body interaction, so it can displace the GABAergic. That was from a group in China, though they have published recently in Cell Report a few years ago. So they show this synaptic displacement of GABAergic synapse is able to dampen neural activity and is able to reduce the seizure they use developmental seizure model. So yes, I mean during development it's a different it's a really important question, but there's different context. So interestingly, the microglia can migrate much more than like in adult microglia. So adult microglia, the cell body not moving much, but mostly processed. But during development, microglia cell body can move. So I assume there was microglia synapse interaction is probably different as well. But that's really important question to directly test that. I don't think there is in vivo imaging evidence there, yet.
Okay, we have two more questions, and then I will make the last one. First from Ali, do you consider NMDA receptor-dependent ATP release from principal neurons a homeostatic or a pathological response to what the microglia react?
Yes, I think I will largely agree. the AMD-dependent ATP release and microglia interaction probably is more pathological. So I didn't get a chance to go like more details in that study. Early studies, we actually titrate the glutamate AMD receptor, glutamate AMD ligand like concentration, and we have to use high glutamate to induce the phenomenon. So really suggests that this could be more related to pathological context. But as I mentioned, in the physiological context, when people study how microglia can sense the synaptoplasticity or regulate memory, so they may have something to do with that. But in our studies, we found AMD receptor dependent this interaction mostly need a higher concentration of glutamate. So presumably more related to the pathological context.
Now, the next one is from Hoyt Dubey again. Does this GABAergic synaptic shield is specific to perisomatic buttons, or do you expect this to be a general mechanism for other GABAergic inputs that are targeting, for example, axon initial segments or dendrites?
Yeah, that we haven't go to that detail yet. But if we look at we actually image the cell body is relatively easier, but the dendrite we found that during the emergence they largely can recover, like not increase interact with the dendrite. So that's why we focus on soma. But it's a great question. I mean, there's even different type of GABAergic synapse, right? There we mostly used like VGAT in the beginning, but later when we do in vivo imaging, we use PV. We use virus to label PV neurons, so the PV button. But how microglia specifically interact, even different type of like GABAergic synapse, and then different location of GABAergic synapse that need to be further studied. Yeah.
There is another question from Trevor Owens, whether the basal microglial state directs how they respond. I mean, that is, do sleep-responsive microglia react differently than the awake-responsive cells? I mean, if you give them the same stimulus?
So, yeah, excellent question. Yeah, that's the kind of active project in the lab to study microglia-neuron interaction in sleep context, because when we do, when we study anesthesia, the next always question is that in the sleep context what happened, and interesting, like there's a recent study use 2-photon, and they use free moving 2-photon. They did show that microglia also increased dynamics and also NE dependent mechanism of very similar to the anesthesia. So, and the sleep context, there is a NE dependent increase of microglia dynamics as well. So we kind of like overlapping what we are doing right now, but we have studied the further mechanism of those interaction and how they regulate sleep and also during sleep deprivation. Yeah, great question.
And I have a short question. I mean, you mentioned that, I mean, you did this study with the contacts from microglia with the excitatory neurons. Did you check the inhibitory neurons? Because in the inhibitory neurons, the GABA synapses are in very specific places, like in the peroneuronal nets holes. So I wanted to ask you if you check these inhibitory neurons as well.
Yeah, not right now, but that's definitely the whole field is moving to that direction, right? So because last 10 years or 15 years, now it's no hanging fruit, so we mostly focus on PMR neurons. But now recent studies already try to target different type of neurons, like GABAergic neuron, like dopaminergic neuron, and Schaffer's group is doing right now. So even serotoninergic neuron. Science group. So I think that's the next steps, right? To try to go more in more details about different type of neurons, different type of synapse, and even different location. So yeah, we haven't studied in a GABA neuron, how microglia will sense this GABAergic input to GABAergic neuron. So we haven't done that. So most of what I show is target excited neurons, so use chemokines 2 as a driver to label the excited neurons. Yeah, great questions.
Yeah, thank you. That's all the questions we have for today. Thank you very much for the talk. It was very beautiful. I saw some question on the chat that they were asking for the recording of the talk. The recording is in the Chucarro website. It's a European Microglia Webinar Series. We can post it on Twitter later, and you can get all the talks there in our website. So thank you, Dr. Wu, for your talk. I don't know, Rosa, if you want to close the session.
Yeah, no, I just want to thanks again, Long-Jun, great. And I want to remind everyone that now we have a summer break and we will come back in September. So don't connect in 1 month because there will be no seminar. But in September, we are very much looking forward to see all of you. Bye. Bye-bye.
Yeah, thank you very much. If you have questions, you can send me email. Thank you, guys. Yeah, bye.
Bye.
Bye.